So we've already talked about synapses, right? We've talked about synapses in a number of different ways. So we talked about synapses in lab, about maybe the third or fourth week of the semester in the nervous tissue lab. And we also talked about synapses in our tissue lecture. We talked about synapses a little bit in the muscle lecture. And we talked a little bit about synapses last week. And so today we're going to really get down into the details of the types of synapses and how they're going to work. And so remember that as a signal is coming from one point in your body to another, whether it be from your brain down to a muscle, or something like a temperature sensor into your spinal cord or up to your brain, that signal can only move down a neuron or down an axon until that axon ends, until that neuron runs out. And then that signal has to be passed from one neuron down to the next. And that's going to be through a synapse. The synapse is the gap between the axon of one neuron and the dendrite of the next. And when an action potential reaches the end of an axon, we've seen this. It's going to cause the release of a neurotransmitter. It's going to come out of those vesicles, right? Vesicles are going to merge with the plasma membrane, release a chemical messenger into the synapse, and that's going to cause the dendrite of the next neuron to fire and then cause the next action potential to move on to the next neuron, to the next, to the next, to the next. But remember what one of these neurons looked like. Most of them didn't have just one dendrite. There were a whole bunch of them. And so for every little dendrite that you have, you're going to have a synapse. And so each neuron is going to have many, many synapses that can cause that neuron to fire. So if we have a synapse, so imagine we have an axon coming down, and then we have the dendrite of the next neuron down here. And then the gap is going to be our synapse. And the signal is going to be coming down this axon into the dendrite here, and then moving down that dendrite. And so the signal comes down this one into this one. So it hits this one first. And so we call this neuron the presynaptic neuron. It's in this neuron before it hits the synapse. And the one after the synapse is going to be the postsynaptic neuron. So this one, the presynaptic neuron, is going to be the one that releases the neurotransmitter. And the one down at the bottom here, the one that's going to receive the signal, is going to be receiving the neurotransmitter. And so the receptors for that neurotransmitter are going to be here. The vesicles will be in here. It'll release the neurotransmitter into the synapse. It'll get bound by the receptor here, and the action potential or the signal will continue.
What is the ACH on the plane?
It's going to have a whole slide for you. At least one slide. So the majority of synapses are going to be these chemical messenger synapses. Some of them, though, are called electrical synapses. And so these are not going to have an open space like this. This is going to allow the electrical current from one neuron to be transferred directly into the next. One place we've talked about this before was in cardiac muscle. Why did we say we needed electrical synapses? Why did we need electrical signal to get from one neuron to another in the cardiac muscle?
So they all go at the same time.
So they all go at the same time, right? We need the heart to pump at the same time. If one side contracts 30 seconds before the other one does, it's not going to work very well. It needs to work in unison. And so this was actually a gap junction. So a gap junction, remember, is essentially a pore from one cell into the next. And so if we have a gap junction, and we have essentially essentially one neuron like this, and the next one touches it, but there's a gap there. The signal is just going to come right down this one, into the next one, and just keep on going. And so yes, a neuron can only be so big. It can't go on forever. It has to end and pass on to the next. But the signal can continue in this case. It's just going to go from one straight down through. And that's going to allow it to be much quicker. Electrical signals move quickly. In a chemical synapse, you have to induce the vesicles. They have to release the transmitter out here. They have to be bound by the receptor. The receptor has to activate. The receptor then has to open voltage-gated channels, which have to open to allow the signal to continue. Over here, this is just going to be much quicker. So, you've seen a picture that looks much better than my drawing, right? At the end of the presynaptic neuron, there is the knob. We saw this in lab. You know what this looks like. And these vesicles, there are some that are here, and then there are going to be some of them that are more or less already on the plasma membrane. They're ready to go. As soon as the signal gets here, they're able to release. And so these are the ones that are going to release first. These are more or less just in reserve. And so once these release, these will take their place. A lot of these chemical messengers will get reabsorbed back up here, and they'll go into the new reserve vesicles. And so it's just a cycle. It's a recycling process. This is, remember, the postsynaptic neuron is going to have these receptors, and those receptors are proteins. They're going to function very similar to the receptors we had in our muscle. And so they were ligand-regulated or ligand-operated ion gates. And so when the chemical receptor binds to this, the chemical messenger binds to that receptor, they're going to open and they're going to allow ions to pass through. This is the, I suppose, better version of my drawing, right? This one you can actually read. But we have the synaptic knob down here. We have vesicles that are bound to the membrane, that are releasing chemical messenger. And then we have the reserved vesicles up here. We have the synapse down here with the loose chemical messenger in it. The postsynaptic neuron, we have the receptors that are down here. So up until this point, we've more or less talked about these chemical messengers in generic sense. We haven't talked about specific neurotransmitters and what they do. So for the most part, we're going to be able to take these chemical messengers and put them into 4 categories. There's going to be acetylcholine, which is going to be a category by itself. There will be amino acids, monoamines, and neuropeptides, and we'll talk about each one more in depth. So like I said, acetylcholine is unique. It is its own category. There is nothing else in its category. It's a combination of 2 molecules, acetic acid, which is like vinegar, and choline. Next to the amino acid neurotransmitters, these are going to be simply amino acids. What in our body was made of amino acids? Proteins, right? Remember, amino acids come together, form a long chain. You get a peptide, and then you get a polypeptide, and you get a protein. And so these are going to be amino acids, like glycine, glutamate, aspartate, are the definition of amino acids. Those are some of the amino acids that get incorporated directly into our proteins. And then there are some that are essentially derivatives of those amino acids. Next category are monoamines. These are very similar to amino acids, but they've removed the acid group. Remember that an amino acid is called an amino acid because it has 2 main parts. There's the amino part, which has the nitrogen, and then there's the acid part. And so a monoamine is an amino acid where they've removed the acid group, leaving just the nitrogen part of that molecule. And so these are the catecholamines, which include epinephrine, norepinephrine, and dopamine, which are messengers you've probably heard of, right? Also in this are histamine, ATP, and serotonin. Which of these doesn't seem like it belongs?
ATP.
ATP, right? We've talked about ATP a lot. And in none of those cases did we talk about it being a messenger. But it actually has another function. It can be used as one of these synapse messengers. And finally, we have neuropeptides. These are a small chain of amino acids. You have at least two, so it's not just one. If it was just one, it'd be the amino acid group. But here we have at least two, and they go up to about 40 amino acids. So a 40 amino acid molecule is pretty big, but it's not gigantic. It's still small enough to be passed around pretty easily. Big proteins like these receptors are going to be much larger than 40. These neuropeptides are going to be stored in granules that can be secreted, and these include cholecytosinin and substance P. These are not as common as these, right? You've probably heard of all of these. You've probably never heard of either of those. So we've also seen that some of these messengers can open channels that are excitatory. We've also seen ones that are inhibitory. Remember in our muscle, when we opened a sodium channel, what happened? It caused it to fire. But we also saw in an excitatory tissue, if you open a channel that allows chloride in, what was the charge on the chloride? Negative. And so in our action potential, when it fires, which direction does the charge go? Does it go positive or negative? Positive, right? You have this positively charged sodium that's going to come in. If you let negative ions in, you're essentially lowering the floor. You make it harder for that action potential to fire. And so some synapses are going to feature neurotransmitters that are excitatory. They're going to allow the positive ions in. Others are going to be inhibitory. They're going to allow negative ions in. In a lot of cases, they may not directly be inhibitory, but they're going to make it harder for the positive, for the excitatory process to happen. And in some cases, the neurotransmitter may actually be the same in both cases. What the difference may be the cell that's receiving it. One cell may receive a neurotransmitter and get excited because of it. Another one may receive the same exact neurotransmitter but get inhibited by it. Remember that a lot of these processes are regulated by our ligand-gated ion channels, but some of the receptors are actually not going to be channels themselves. They're going to get activated, and then they're going to either turn on or off other channels that are next to them. And so we have three types of synapses. We have excitatory cholinergic synapses. We have inhibitory GABAergic synapses, and excitatory adrenergic synapses. Of course, we'll talk about each one. So these are the cholinergic synapses. So remember, these are excitatory, right? So these are going to use acetylcholine. And in most cases, people don't like writing acetylcholine over and over and over again. And so it gets abbreviated ACH, where the H is lowercase. So that's acetylcholine. So imagine we have a signal coming down a neuron. It's going to reach the synaptic knob. It's going to open voltage-gated channels, that are going to allow calcium in. When calcium comes in, it's going to cause the vesicles to release the chemical messenger into the synapse. That chemical messenger in this case is acetylcholine. And so it's going to come across, it's going to bind to the receptors in the next neuron. That's going to open up, allow sodium, potassium to do their thing. Sodium coming in is going to cause these postsynaptic neuron to become depolarized. So we're going to get a whole big influx of positive charge, and that's going to cause the action potential to continue. So an acetylcholine synapse, a cholinergic synapse, is essentially the prototypical synapse that we know. So that's what we were just talking about. The signal's going to come down. It's going to cause this calcium channel to open, which allows calcium to come in, causes the release of acetylcholine, activates these channels, which allows sodium to come in. And the signal has been passed from one neuron to the next. Next type of synapse is a GABAergic synapse, and these are inhibitory. A GABAergic synapse, just like the name makes it sound like, is going to use gamma-aminobutyric acid, which gets abbreviated GABA. And so in this case, when the nerve signal comes down, instead of releasing acetylcholine, it's going to release this GABA. But the GABA receptors are chloride channels. And so when the GABA gets released, the chloride channels are going to get open. Chloride is going to come in. It's going to make the next postsynaptic neuron more negative, making it harder for it to fire. It's possible you might have cholinergic receptors and GABA receptors here. But if you send the GABA, you essentially turn this off. If you then turn around and send the acetylcholine, it's not going to be able to excite it. So I found there wasn't a diagram of this in the slides. I actually found this. It was in German. It was the simplest one that I could find. It was what I wanted to show, so I kind of blocked out some of the German, but my looks like my white blocks kind of moved on me. You can probably action potential, right? I think this one was like wieder Hausnatzen or something like that. I don't know. German's such an awful language, right?
Sounds fun. Or really aggressive.
Yeah, if you ever want to think of it, look up the YouTube video, not now, of the comparison of languages. It's like they go through English and French and Italian and all these languages, and Spanish, and these words, and they all sound just so nice. And then you got the German guy wearing lederhosen who says the German word, and it's just awful. Right? Yeah.
Oh, Russian.
Why? I don't know enough Russian.
It's pretty rough.
I just know. So in lab, there's a centrifuge that I use. And so imagine there's a lid on a centrifuge. It's just a little circle lid. Okay? And you just kind of pop it on before you spin your centrifuge. And up here, it says caution. And it says something like, make sure it's snapped. And then down below, in one word, in German, it goes like this. It's one word. There's no spaces. I have no idea. I have no idea. I'm half German. So, action potential is going to come down. Calcium is going to come in just like before. It's going to cause the synapse to release our messenger, but the receptor is the chloride channel. And so it's going to allow chloride into the chloride canal or kennel, right? Channel, but my text box knocked the L down to the next line. Can't win. Oh, I did love that. Wiederhof. Wiederhof. But the word comes to like there. It's always, well, what is it? I apologize to you that came here to learn. It's like, Wiederhof now. put that on an exam. Do not say, leave your option now. Okay. Luckily, the chair left before I had to go through that slide. I couldn't, that would have not been funny.
I thought you just gave up.
I gave up on life, or?
No, she gave up on
Oh, she gave up on me.
No, I'm learning the stuff.
Oh, she teaches this.
I know, that's all.
You're giving up on her, right?
Give up on my joke.
So, the next are adrenergic synapses. So adrenergic synapses are excitatory, again, and these are going to use norepinephrine, which is also called noradrenaline. Norepinephrine is going to get abbreviated NE. These are that different type. So the receptor is not going to be a channel itself. It's going to be attached or next to a channel. So it's going to have to pass that message on. And so we call this a second messenger system. And so the receptor that's going to receive the norepinephrine is going to create a second chemical messenger. And so it's going to create a second chemical messenger inside the postsynaptic neuron. And these chemical messengers are going to activate other proteins there that are channels that are going to open up and allow the ions to come through. So the most common example of a second messenger is cyclic AMP. So AMP is just like ATP, adenosine triphosphate. This is adenosine monophosphate. So there's only one phosphate instead of three. But you can actually take that molecule, and it's kind of it's not a long molecule, but imagine that's AMP. You just kind of rotate it around, react the two ends together, it's now a circle. So it's cyclic AMP. And so the receptor is going to bind the norepinephrine. It's going to make cyclic AMP, which is going to float around and activate these other channels. This is slower, right? The chemical messenger is not going to directly open a channel. It's going to activate another protein which has to create cyclic AMP, which has to then move over and activate the other channel. But there has to be a reason that these exist then, because it's not normally a good thing for there to be a delay. The benefit of these is that you can amplify a signal. If you don't use a second messenger system, one of these messengers can open one channel. But with a second messenger system, if you activate the receptor, one messenger molecule can activate one receptor, which can then make an unlimited number of cyclic AMP molecules. And so it's possible that one chemical messenger can activate one receptor, which may activate 100 channels. And so you get these very large amplification signals. And so it allows a very, very strong signal, some more than you can accomplish with a direct channel opening. So this is this process. So presynaptic neuron is going to release norepinephrine in the synapse. It's going to get bound by the neurotransmitter receptor. So here's norepinephrine binding to the receptor. It's going to activate something called the G protein. These are beyond what you need to know. But it's going to activate something called the G protein, which is then going to activate the other protein that makes AMP, adenosine cyclase. So this is then going to make cyclic AMP, which is a whole bunch of things in our body, one of which opens some channels. So you just need to know this process, right? The neurotransmitter is going to bind, it's going to activate some other proteins that then open up our channel. But this all has to stop, right? If I touch the board and the touch receptors in my hand send a signal to my brain that I'm touching the board, when I take my hand off, it needs to stop. Otherwise, it's a one-time use hand, right? And so the first thing you touch, you're going to be feeling it the rest of your life, right? So hopefully it doesn't hurt. Basically, this has to stop. And so we have to have a way to turn these signals off. One way is that the presynaptic neuron can just stop releasing neurotransmitter. This neurotransmitter, some of it's going to get used up, some of it's just going to float away. And so eventually, there won't be any left in the synapse. If there's none left in the synapse, this one will stop firing. The neurotransmitter, when it binds to the receptor, it's only going to stay there for about a millisecond. That's a thousandth of a second. So it's not going to stick and stay. It's going to stick, and then it's going to come off. And so even the ones that do bind are going to diffuse into the extracellular fluid. They're just going to float away. One of the purposes of the astrocytes, remember that's one of the big major types of neuroglia, is to grab onto these neurotransmitters and reabsorb them. That's going to take them out of the synapse, stopping the signal. The synaptic knob here can directly reabsorb some of the neurotransmitter with endocytosis. Remember, endocytosis uses things like vesicles. It just kind of takes back in. It's the reverse process. So it's going to create vesicles inside the cell by taking in the neurotransmitter and extracellular fluid out here. Acetylcholine and some of the other transmitters also just break down over time. Some of them are unstable, but break down on their own. Acetylcholine has an enzyme that floats around in the cleft, in the synapse itself, is going to break down acetylcholine. And so some of it's going to diffuse away, some of it's going to get absorbed by neuroglia, the astrocytes, some will get reabsorbed by the presynaptic neuron, and some of it's just going to get broken down. That's our synapses. Does that make sense? At least enough to get you to next week, so you can look at it again, right? All right. So the last thing I'm going to talk about for the general nervous system lecture is neural integration. And neural integration is well beyond what we are going to learn. Okay, we need to cover what it is and the very, very, very basics of it. But we are not going to get into it at all. Neural integration is the process of thinking and memory, right? If you were going to imagine or name a part of our physiology that we don't, as humans, understand, it's those things. Memory and learning, we don't have a very good idea of how that works whatsoever. And so even the best people can only give just kind of a surface explanation. So we're not going to get too deep into it. Essentially what this is, when we say integration, we're going to be taking a whole bunch of signals. Visual signals, touch signals, all the different senses together. All those signals are going to come together in our brain, and our brain is going to make one idea out of it. So that's what neural integration is. It's going to make that idea, it's going to figure out what to do with it, and it's going to store it in your memory. The whole idea, as the most basic part, is based on postsynaptic potentials. So a postsynaptic potential is just the action potential that happens down here, right? This is the postsynaptic neuron. This is a potential in the postsynaptic neuron. But these are not going to be above threshold. These are going to be below threshold. So it's not going to cause that whole action potential to fire off. This neuron's going to receive it, and in one way or another, it's going to remember it. And so it's going to receive a pattern of subthreshold signals, and it's going to know when I saw that pattern, this is what was going on. And so later on, if we think about what something looked like, if I look at Gibby, I see, my brain says that is Gibby, that is what Gibby looks like. I then look over here, and I say, what did Gibby look like? My brain says, well, when I was looking at Gibby, these signals came from my retina into my brain. So let me replay those signals again and see what Gibby looked like. It's crazy, right? But that is neural integration. It's postsynaptic potentials. So remember, this is going to be below threshold. It's going to be the same process. Sodium is going to come in, but it's not going to be enough to hit threshold. So an action potential is not going to go. This can be inhibited by some of these inhibitory chemical messages, right? So if we have something that's going to open a chloride channel, it can block memory. If we have something that allows potassium to get out of a neuron, it can also block this memory and learning. This is sub-threshold potential. So remember their voltage, we start at -70. With an action potential, we come up, we hit threshold, and then it spikes, and it comes back down. Here, we never reach it. And so you get just a series of these sub-threshold potentials that are going to cause memory. So here, this hyperpolarization, this is going to be an inhibitory signal. And so we're going to be able to make a combination of these and these to make a memory. It's just, that's as far as we're going. Because you can talk about that your entire life and never really know what you're talking about. All right. Questions about this lecture? I think it's probably not quite time for, right, we'll talk a little bit more about the next one before we take our break. Anybody remember his name?
Brain.
And why do I have a mouse?
Brain.
That's brain, right? Everybody knows brain?
Pinky was my
It's brain and spinal cord. There's no pinky.
But Pinky
So I had to cut pinky out. Pinky doesn't belong here. Pinky's out trying to take over the world. Now, how many people have the song stuck in their head? How many people have no idea what we're talking about? At the break, look up Pinky in the Brain. How many people have no idea who he is? Yeah, this is from the old Teenage Mutant Ninja Turtles. The real Teenage Mutant Ninja Turtles. Not the fake ones that they got going on. The real ones. They're real. Don't tell me they're not real. Okay, we got to talk about the brain and the central nervous system. Your slides are all black and white. I've gone through and color-coded the slides based on whether we've covered them or not. And so the green ones that we'll have up here are slides that we have gone over. If you see blue ones, those are slides we have not gone over. You are still responsible for all of them. It's just we're not going to spend a lot of time on the ones we've already gone over. So this is our brain. We had a couple directions we talked about when we got reference to the brain. Remember that rostral is forward, is towards your forehead. And so that's going to be here. And towards the back is caudal. Rostral and caudal. So when we talk about different parts of the brain, sometimes we'll be saying that one is rostral compared to the other. Brain is about 3.5 pounds. Probably bigger when you leave out of here, right? That actually doesn't get bigger when you leave. Not at all. So the brain had three main parts. We know this. There was the cerebrum, which is what we think of as the brain. There's the cerebellum, which is still fairly large, but it's separate down here. It's a separate entity. And then there was the brain stem, which is at, remember, the very top of the spinal cord. The spinal cord is coming up. We've got the brain stem. It kind of extends up into here. So the brain stem is going to be what is left if we delete the cerebrum, we delete the cerebellum, and we delete the spinal cord. Whatever is left is going to be the brain stem. We had a number of lobes. There's another figure that's better for lobes. But remember that the cerebrum has two hemispheres, and it's separated by the longitudinal fissure. On the surface, we have ridges and we have grooves. The ridges were our, it's not even labeled on this one. What was the ridge called? After the G?
Gyrus.
Gyrus. Yeah. The plural form is gyri or gyri. I honestly don't know which one's correct. Just gyruses. And then you have the sulcus, which is the grooves. And so the gyrus is the ridge. The sulcus is the grooves between the ridge. The two hemispheres are connected. You can't see, it's not on the surface, it's kind of buried. The corpus callosum is a bundle of nerves that's going to connect one side to the other. That's what's going to keep those two hemispheres from literally just falling apart in your head. The cerebellum also has two sides, and it also has gyrus, sulcus, and fissures. The fissure between the cerebrum and the cerebellum is called the transverse cerebral fissure. That was something, I think that might have been on the lab exam. We had our brain stem, which was made up of the diencephalon on top, which has a number of other parts we'll talk about. It has its own slides coming up. Below that was the midbrain. There was the pons, and there was medulla oblongata, which we'll talk about later today and then also next week. This was kind of a number of things that we went over. I'm not going to take too much time on this, but this slide is nice because it has a whole bunch of stuff on it. And then you've got the real picture. Nice, hard. Easy, hard, right? When the brain surgeon goes into your head, unfortunately, it doesn't look like that. Right? I suppose that's probably why the brain surgeon is getting paid what the brain surgeon is getting paid. Because I look at that and I say, whoa, I have no idea where one thing ends and the next one starts. You don't want me cutting into your brain if I say, I don't know where one thing ends and the next one starts, right? I'm okay with that. I'm not okay with that. So these were our lobes, right? We had the frontal in the front, kind of the parietal, top and back, occipital in the very back, cerebellum was down here, and the temporal was kind of on the side by our temples. We had gray and white matter. What is the difference between gray and white matter? Why is one gray, one white? Amount of myelin, yes. Which one has more?
Light.
White. Myelin is white in color, and so white matter has myelin, gray matter does not. We had 3 different meninges. We had the duramater, what was below the duramater? Think of a spider. Arachnoid mater. And then below that was pia or pia, depends on whether you want pie or not, if you're hungry. So the duramater was the outside, it's the one on the outer, the most superficial meninges, meniscus, whatever. This is the one that had the folds that you had to memorize. This is the one that was on the lab exam. And so we had the falx cerebri, which goes between the cerebral hemispheres. We have the tentorium cerebelli, between the cerebrum and the cerebellum. And then we have the falx cerebelli, which divides the left and the right halves of the cerebellum. Below that was the arachnoid and the pia. The arachnoid mater is below the dura mater. Below the arachnoid mater was the subarachnoid space. On top of the arachnoid mater, so between the dura mater and the arachnoid, was the subdural space, sub under dural, dura, so under the dura mater. And then the pia mater was very, very thin. It was basically like shrink wrap on the brain. It's very thin, it follows all the little grooves. And so here we saw, we had dura mater, we had the arachnoid mater with the subarachnoid space below it, and then we have the pia mater, which is right on the brain. We also have the ventricles, which I think people know pretty well. The ventricles are pretty straightforward. We have 2 lateral ventricles. We had the third ventricle, fourth ventricle down here, and the third and fourth were connected by the cerebral aqueduct. Again, wow, right? Not as nice. Apparently the cerebral aqueduct is not aqua in color. Who knew? Thank you. So ventricles are filled with cerebrospinal fluid. The cerebrospinal fluid was made by the choroid plexus. That was on the floor of the ventricles. So the bottom part of the ventricles is the choroid plexus, and those are blood capillaries that are going to produce the cerebrospinal fluid. Why would blood capillaries make CSF? What is CSF? What is it made out of?
Blood plasma?
Blood plasma. It's blood plasma with a bunch of stuff taken out of it. And so, if you're going to start with blood plasma, you might as well start with blood. And so the blood capillaries have a constant flow of blood, so they can filter things out of that plasma to make the CSF. So the ependyma are neuroglia cells that are actually going to make it. And so they are on the capillaries. So the vast majority of our cerebrospinal fluid is made up in the lateral ventricles, and then gravity is just going to let it flow down into the third, through the cerebral aqueduct, into the fourth, and then from the fourth into the spinal cord. And most of it's going to get reabsorbed down in the spinal cord. Cerebrospinal fluid had a number of purposes. One, it kind of floats our brain. It protects our brain. If I move my head left and right, it's going to provide some cushion between my brain and my skull. And so it's going to kind of absorb some of that movement to protect it. And it's also going to kind of create an environment around the brain that's conducive for the brain function. So it's going to put the ions in there that need to be there. It's going to collect the waste that comes out of our brain. Why would there be waste coming out of our brain?
From our brain cells? cells?
Right. Our brain is made of cells. Cells make metabolic waste. They break down glucose, they break down ATP, they make a lot of metabolic waste. We need to get that waste away from our brain. That was another just kind of summary diagram, and now we've got blue. So we did not talk about blood supply to the brain. And so we kind of just started with there's blood in the choroid plexus, and that's going to be used to make cerebrospinal fluid. But we need blood in our brain other than for the purpose of making CSF. We need a lot of blood in our brain. Of our body, our brain is only about 2% of our weight. But 15% of the blood in our body is in our brain. So in our brain right now, going in and out of the brain is 750 milliliters per minute. That's a lot. So just a standard water bottle that you buy is 500 milliliters. So 1.5 of those bottles, probably roughly equal to about a 20-ounce bottle every minute. That much blood going in and out of your brain every minute. Well, at least hopefully it's the same going in and out. Otherwise, so neurons use a lot of ATP. We have all of that sodium rushing in every time an action potential goes through. But in order for the next action potential to go through again, we have to pump all of that sodium back out. And remember, to get that sodium back out requires pumps. And pumps use ATP. And so our brain is going to go through a lot of ATP. And in actuality, if we lose blood flow to our brain for 10 seconds, we can lose consciousness. And so our brain is going to shut off the non-required functions. We don't have to be conscious to be alive, right? We have to breathe, our heart has to beat, our organs have to work for us to be alive, but we don't have to be conscious. So after about 10 seconds, our consciousness is going to shut off. After only 1 to 2 minutes without blood flow to our brain, we can basically stop working. So all those important functions are going to stop working. And after 4 minutes without blood to your brain, you're going to have irreversible brain damage. Because our brain is going to just fly through the ATP that's in there. My heart stops beating, and my brain is still working. I'm going to go through that ATP, it's going to be gone, and eventually it's not going to have enough ATP to survive. So those brain cells, those neurons are going to start dying. So we need a lot of blood getting into our brain. But if there is one part of our body that we need to protect above all else, it is our brain. And so we need to be careful about what gets in there. We can't just let anything get in. And so what we have, what your textbook calls the brain barrier system, most other places are going to call it the blood-brain barrier or the BBB. Okay, same thing. This is going to prevent some substances from getting into our brain. It's also going to prevent some things from getting out of our brain. There are going to be bad things in our blood that we don't want getting into our brain. What could those things be?
Bacteria.
Bacteria. What else? Say it again? Viruses, yes, definitely. Bacteria, viruses. What about things that aren't alive? What's something that can kill you that is not alive? But very small, not like a bullet. Blood-brain barrier can't stop a bullet.
Toxins.
Toxins, yes, thank you. So we have chemicals that we do not want in our brain. And so our body all the cells in our body are making that metabolic waste. It's going in the blood. If we can prevent that waste from getting into our brain, we're going to be a whole lot better off. It needs to be in our blood because it has to get to our liver, it has to get to our lungs, it has to get to our kidneys. But if we can prevent it, create a wall that says no further, you can't go past this, turn around and go back. That's what the blood-brain barrier is going to do. And so there are two ways that blood is going to get into our brain. And two places that these bad things could get in. There are the capillaries themselves that are carrying blood to our brain, but then there is also the choroid plexus. And if we prevent the virus from getting into the blood in our brain, though we let it into the cerebrospinal fluid, we didn't really help ourselves very much. So we need to keep them out of both places. And so the blood-brain barrier contains cells that have tight junctions. So if we look at a normal blood vessel, so imagine this is a blood vessel, and we have blood that's flowing through here. This actually isn't a nice solid wall. These are epithelial cells and smooth muscle cells, one next to another, next to another. And so there are actually little breaks. And so things in your blood can leak out. That's on purpose. That's how these things work. But in our brain, we don't want that to happen. Because things like the viruses and bacteria can then leak out. And so these cells here have what are called tight junctions. We learned about these in our cell lecture. Probably don't remember what they were. But these tight junctions were where the two cells were right up against each other. And so they basically share a wall. So there is no space between those cells for liquid to get through. And so if we have tight junctions here, the water, the blood, can't get through between cells. It has to go through a cell. In order to get out of the blood vessel into the brain, it has to go through a cell. If it has to go through a cell, that cell can say what gets through and what doesn't. If it's going through a gap is nothing. A gap can't stop something from getting through. If it goes through a cell, it can prevent certain things from getting through. And so the tight junctions are going to actually be induced by astrocytes. So astrocytes were neuroglia. And so imagine we have our cells that normally would be making our blood vessel. So the blood's flowing through here. We have our gaps here, just normal cell-cell connections. So the astrocytes, let's call that an astrocyte, they're going to reach down and they're going to touch these cells and they're going to make these cells fuse together. And so there's nothing special about these, it's just they have the astrocyte that tells them that they need to fuse together and form these tight junctions. So some things are going to be able to get through this very well. Some things are not. So obviously we need water to get through. We also need glucose to get through. Lipid-soluble substances, so things that dissolve in fat or oil, are also going to get through. So oxygen, carbon dioxide, alcohol, those are lipid-soluble substances, so they are going to be able to get through. Caffeine, nicotine are able to get through also. And then anesthetics are able to get across the blood-brain barrier. If they didn't, they wouldn't work very well. some things are able to get through a little bit. So sodium, potassium, chloride, so the things that really make our neurons function, they need to get in there, because some of it's going to get lost over time. We need to be able to replace that. But we also don't want too much getting in. We want our brain to be able to control how much sodium, potassium, and chloride are there. Urea and creatinine are waste products that need to be able to get across. If they can't, these waste products can't get out of our brain, go the opposite direction down into the blood vessel. Okay. That's blood-brain barrier. Easy enough, straightforward. Okay, so we talked about different parts of the brain. Let's stop. Let's not stop, but you know. Let's take a 6-minute break. So 7:05 on my watch. Right? You can all see my watch.
The clock also works.
Oh, okay. Then we'll go 7:06 on that clock.
She was a new student first day in class, and so he gave up trying to learn it.
I don't know if no one has noticed, whoever just stood there to say anything. I'm wearing a fishing Santa hat.
Actually, I was going to ask if there's like lights on
I
it.
figured why not. Christmas in July, right?
That's right.
Did your phone survive that call?
Yeah, but I just got my phone and cellular crack. Just like,
Oh, bad. I thought you were going to pick it up and blast it.